So the topic that I'll be explaining today is going to be PCR, or polymerase chain reaction. Now, what is a polymerase chain reaction? So for medical purposes or for research purposes, whenever a small strand of DNA or a gene is taken, sometimes we need to amplify it. Now the word amplify means to multiply. So when we get DNA samples or a certain segment of DNA or a certain part of a gene, it will not always be in huge quantity, and in order to conduct experiments or to do medical tests or to do any other type of forensic tests, it is very important that we amplify this DNA sequence or gene into multiple copies so that they can be screened in gel electrophoresis or any other similar technique. So the main concept of a PCR or a polymerase chain reaction is to amplify a DNA segment or a part of a gene. Now for this purpose, special machines known as thermal cyclers, or what we call PCR machines, are used. So small tubes known as PCR tubes are inserted into the thermal cycler, and then the machine is started, which is then continued for about 20 to 40 cycles. Now we will come back to these cycles later on, but before we go into the cycles, it is very important to know that these cycles or the number of cycles that we do depends on the amount of DNA that we have in the first place. So if we have very less amount of DNA in the first place, we may need to do 40 cycles, and if we have a decent amount of DNA already, then we may only need to do 20 cycles, which is also not fixed. These can vary even lesser than 20 cycles and even more than 40 cycles. Now going away from the number of cycles that we are going to take here, the PCR tube that we will take is going to contain what we know as a master mix. Now what is a master mix? Master mix is made up of various components. First of all, the very first component of the master mix is going to be the DNA template. Now what is the DNA template? DNA template is the template of DNA which we are going to use to amplify. That is, it is our target DNA which needs to be amplified or which needs to be multiplied. So obviously that is the very first thing that we are going to need in the master mix. Secondly, we are going to need primers. Now what are primers? I'll be getting into more detail in the primers when we come to the second step of PCR, which is annealing, but for now it is just know that there are two types of primers: forward primer and a reverse primer for the two different strands of DNA, which I'll be talking about later. Third thing that we need after the primers is DNA polymerase. Now I don't think it's right to write DNA here, because most commonly used DNA polymerase is Taq polymerase, something that you may have already heard of. Now what is Taq polymerase? Taq polymerase is a DNA polymerase that has been extracted from Thermus aquaticus, which is a bacteria that thrives in high temperature conditions, which is about 80 degrees Celsius. And the reason why we use DNA polymerase is because I will explain that in the first step of PCR, which is denaturation. Now the fourth main component of the master mix is going to be DNTPs. And what are DNTPs? DNTPs are the deoxynucleotide triphosphates. Now these triphosphates will be adenine, guanine, thymine, or even cytosine, AGTC, any of these. Now the last component of the master mix is going to be a buffer, and this buffer is a very important in order to maintain the pH and every other optimum condition for the master mix, and for the DNA to survive in it, and for the DNA polymerase to survive in it, and for the DNTPs to survive in it comfortably. That is, it provides optimum condition for all the components of the master mix so that it can be done and properly used in the PCR machine. Now coming to the main steps of PCR. So PCR is divided into three very simple steps. which includes first of all denaturation, second will be annealing, or we also call it primer annealing because it is the primer that is getting annealed to the DNA template, and lastly we come to the extension, which is the extension of the DNA template. Now let us start with the first step in PCR, which is denaturation. Denaturation, as we can tell from the name itself, means to denature the DNA. So the DNA template that we will be taking will be double stranded, right? And because it is double stranded, it is going to have hydrogen bonds. In our body, we have helicase enzyme, which is used to break these hydrogen bonds for the DNA strand to be separated during the process of replication. Now, unfortunately, because we cannot use the helicase enzyme for performing PCR, what we do is we use temperature as a replacement for the helicase enzyme. Now, the temperature will be about 94 to 95 degrees Celsius. So at this temperature, what happens is that the hydrogen bonds between the DNA template will break down, and the DNA template will be separated into two different strands. One will be ranging from 5' to 3', and the other will be from 3' to 5'. That is a forward strand and a reverse strand. It is also important to note here that this process has to be done for about 30 seconds to 1 minute, and the reason is because it is very important that the hydrogen bonds are completely broken and the two strands of DNA have moved apart, because in case if the DNA strands are near to each other and they haven't completely broken apart, the bonds will be reformed and the DNA will again become a double strand. Now we are done with denaturation. It is a very simple process. Double strand is converted into two single strands. Now the two strands that were made here will come into annealing. That is the first strand, which ranges from 5' to 3', and the second strand, which ranges from 3' to 5'. Now we are going to have two primers. As I already mentioned, there will be a forward primer that will go from here 5' to 3', and there will be a reverse primer that will go from 5' to 3' on the other strand. So the reason why we have two primers is because the two strands of DNA that we have separated need to be replicated at the same time, because that is more efficient. So what happens is, if we use only one strand of the DNA to replicate, it will take a lot of time to produce the amount of DNA that can be produced using two strands. Hence why two strands are used. Now the biggest question in this is, why do we need a primer in order to replicate or in order to add nucleotides to our DNA or in order to extend our DNA? The answer to that is because the DNA polymerase enzyme that we will be using, or any DNA polymerase, is not going to work on a DNA strand unless and until it is double stranded. So for a DNA polymerase to come and attach to the DNA strand in order to add nucleotides, it has to be double stranded. So the thing is, the DNA polymerase needs a double strand in order to come and attach to the DNA for it to then start the process of extension, that is the process of adding nucleotides in a sequence which is complementary to the DNA strand. So annealing also has to be done at a certain temperature, which ranges from about 50 degrees Celsius to 65 degrees Celsius. Now once again, as I already mentioned, this temperature is highly variable. That is, this temperature will be different for different primers. And how will it be different for different primers? According to the Wallace rule, which states that A plus T into 2 degrees Celsius plus G plus C into 4 degrees Celsius. Right. The answer to this will be the melting temperature. Now this melting temperature is the melting temperature of the primer. Right, this melting temperature is the melting temperature of the primer, and hence it is very important that whoever is making the primer knows the melting temperature, and the temperature that we will use for annealing will be 1 to 5 degrees Celsius lesser than the melting temperature. And this is because we do not want to degrade the primer before it even attaches to the DNA, because then the process will not continue. Hence why the melting temperature of any primer is calculated, and then 1 to 5 degrees Celsius lesser than the melting temperature is taken as the annealing temperature, and then the process of annealing is done. This is a rule that was given by Wallace. And just for extra information, the reason why 2 degrees Celsius is being used here and 4 degrees Celsius is being used here is because A and T have double bonds, whereas G and C have triple bonds, and it requires lesser temperature for A and T to break the 2 double bonds, and it requires more temperature for the triple bonds to break, and hence why this equation has been made in such a way. And using this equation we can get the annealing temperature, which can then be used in order to anneal the primers to the DNA template. Lastly, we come to extension. So what is extension? At this point of time, after providing enough temperature and time, the time also to be noted here is that it will be about 30 seconds to 1 minute. This too is going to vary on the type of primer that is being used, because the primers vary from about 15 nucleotides to 25 or even 30 nucleotides. Once again, this value is highly variable for the DNA template that you are using and for the primers that you are making, and these primers have to be highly specific so that they do not get attached to some other part of your DNA and form copies of DNA that you do not require. Now coming to extension, the DNA at this point in time, the DNA template will be in a condition such as this, along with the primer annealed with it. Now what needs to be done here is that the Taq polymerase enzyme will come and attach here, right? And after the Taq polymerase enzyme comes and attaches to this double strand which has the primer and the DNA template, it will start doing its work of replicating by adding nucleotides. Now from where does the nucleotides come? As we added in the master mix, D and TPs, the deoxynucleotide triphosphates will come and get attached, and just like how the process of replication occurs in the body, in a very similar way the process of extension or the process of addition of nucleotides will also happen here, and this is going to happen simultaneously on 2 strands of DNA. One is the forward primer and the reverse primer on the forward strand and the reverse strand. Extension is done at a temperature of about 72 degrees Celsius. This is quite constant. It is not changed according to different circumstances. Most of the time 72 degrees Celsius is used for extension, and about 1 to 2 minutes are given to this process. Now what happens in this is that these processes, all the 3 processes occur simultaneously, one after the other, and that includes the change in temperature as well. And the reason why we are taking Taq polymerase is because in the master mix, all of the components that are involved in the PCR process are added in the master mix, and because the Taq polymerase is added in the master mix, if any other polymerase enzyme, for example DNA polymerase from the human body was used, it functions properly at 37 degrees Celsius, and if the temperature is raised to about 94 or 95 degrees Celsius like here, this DNA polymerase will become non-functional. It will get degraded because it is a protein. Hence why we use Taq polymerase enzyme, which is extracted from Thermus aquaticus, which can survive in temperatures such as these, so that later in the reaction when we come to extension, the Taq polymerase can get attached to the DNA strand and do its work. Now that is it for PCR. Thank you very much.